This recording will explore some basic concepts related to the control of gene expression. When we think about the control of gene expression, we can identify at least three major levels at which gene expression can be regulated. So on one hand, we can talk about transcriptional control. This would involve having the appropriate transcription factors and RNA polymerase present in order to get transcription going. Even if transcription does take place and a messenger RNA, shown here in yellow, is produced, we can also talk about translational control. So the rate at which translation takes place, that has an impact on overall levels of gene expression. And even once the polypeptides or proteins are produced during translation, we can also talk about post-translational control, because forming the initial protein typically is not the end of the story. Often those proteins have to be modified. They can be activated or deactivated depending on the environmental conditions. They have to be modified before they could become fully mature. So the point is, between going from DNA to a fully functional protein, lots of events have to take place, and those events can all be regulated and, of course, all have an impact on overall levels of gene expression. During this recording, we will explore the lac operon. This is a classic example of gene expression that was worked out in the 50s and 60s, and this represents a classic example of transcriptional control. That is, how genes are transcribed only at the appropriate times within cells. So let's talk a bit more about this so-called lac operon. Before we say more about what lac operon refers to, let's set the stage here. Think about bacteria, for example, E. coli. Bacteria can use lots of things as source of nutrients. One of the things that they can use is lactose. So lactose is the sugar found in milk. So in order for E. coli to use lactose as a source of energy, a few different proteins have to be produced. On one hand, if you notice this green protein toward the left, this permease, this is an enzyme that allows lactose to enter the bacterial cell. Once inside the bacterial cell, an enzyme, galactosidase, this enzyme splits lactose into its individual monosaccharides, namely glucose and galactose. So again, those two proteins, shown in green, they have to be expressed in order to use lactose as a source of energy. So the genes that code for those proteins are referred to as the following. So LAC-Y, that's the gene that codes for this permease, which allows lactose to enter a cell. The LAC-Z gene is the codes for the enzyme beta-galactosidase. This is the enzyme that breaks lactose into its individual monosaccharides. There's another gene that was discovered later, the LAC-A gene, but its explicit role in the lac operon is not entirely understood. So the point is, these genes, LAC-Z, LAC-Y, and then later LAC-A, they are expressed only when lactose is present, only when needed. So the basic question, when are those genes expressed? Well, again, they're only expressed when needed. That is, when lactose is present, and lactose can be used as a source of energy for these bacteria. So work done by two scientists, Jacob and Minode, figured out how this whole process works. And again, these are the folks that figured out this lac operon. And when we talk about the lac operon, lac referring to lactose, operon referring to a situation where we have multiple genes. So here we have the LAC-Z, LAC-Y, and there's this LAC-A as well. Multiple genes that are expressed and transcribed as a single unit. And so they're all under the control of a single promoter, shown here in blue. So here we're looking at the lac operon. Just to set the stage. Again, we have our functional genes, LAC-Z, Y, and A, that are expressed when lactose is present. We have a promoter for those genes. We also have this operator. We'll talk about that in just a moment. And also, if we look upstream, there's another gene I want to mention, LAC-I. I for inhibition or inhibitor. So how do all these units or components interact with each other and allow these functional genes, Z, Y, and A, to be expressed only when lactose is present? Well, let's explore how that works. So what we're looking at here is just a schematic of the lac operon like we just saw previously. So toward the left we have our LAC-I gene. Further toward the right we have our promoter operator and our three functional genes, Z, Y, and A. involved in this whole system. So we also have a few things toward the bottom of the screen. We'll talk about those in just a moment. Let's first of all explore what happens if lactose is not present. So if lactose is not present, as you might imagine, there's no need to express the Z, Y, and A genes. So when lactose is not present, essentially what happens is as follows. This LAC-I region, it's a gene, part of DNA, it codes for this repressor protein. So if no lactose is present, the repressor protein that we see toward the left there essentially makes its way and binds to this region right here, referred to as the operator. So again, if lactose is not present, this repressor protein binds to the operator. Imagine the RNA polymerase that we always need for transcription binding to the promoter. What it's going to try to do is sort of make its way toward the right or downstream to code for the LAC, Z, Y, and A. But if that repressor protein is bound to the operator, the RNA polymerase is stuck. It can't really go anywhere. So as a result, when lactose is absent, the LAC, Z, Y, and A genes are not expressed. What happens then if lactose is present? Lactose, shown here just schematically as a triangle, lactose, if it's present, what it can do is actually bind to this repressor protein. When the lactose binds to the repressor protein, it causes that repressor protein to change shape. So when that repressor protein changes shape, it is no longer able to bind to the operator. So if the repressor can't bind to the operator, the RNA polymerase is now free to bind to the promoter and make its way down along the LAC operon and transcribe the LAC Z, the LAC Y, and LAC A genes. So, again, as a result, if lactose is absent, the repressor binds to the operator, genes are not transcribed. If lactose is present, it binds to the repressor protein, changing its shape, and therefore the repressor does not bind to the operator, and under those situations, RNA polymerase makes its way down, thereby transcribing the functional genes involved in the LAC operon. So that was quick and brief, and also keep in mind that the LAC operon, as I just described, it's much more complex than that. There are a lot more components that come into play that we're just not mentioning at this point, but nevertheless, that gives you a brief introduction to one of the ways in which gene expression can be controlled.